Hello, and welcome to my presentation. I am Jacob Coppolino, and my supervisor is Dr. Susan Glasauer. We operate out of the School of Environmental Sciences at the University of Guelph. The project is on the effect of freeze-thaw cycles on surface soil phosphorus mobility in a Southern Ontario riparian buffer zone. The contents of today's presentation will cover the background and trends of this system, a site description, and then the project is broken up into a field and a lab study with each their individual results, and then a discussion and a conclusion. Lake Erie's interaction with Southern Ontario's agro-ecosystems is being used as a case study. Historically, Lake Erie has experienced phosphorus loading and harmful algal blooms. Recently, this issue has re-emerged, even though the amount of phosphorus applied on agricultural fields has declined. The Lake Erie Action Plan recognizes this, as well as the potential to increase with climate change. It is this potential to increase with climate change is why we are interested in investigating the Grand River watershed's riparian buffer areas and if phosphorus loading could be further worsened under future climate conditions, specifically freeze-thaw cycles. The key driver of this future change is that climate change is projected to increase springtime freeze-thaw cycles in mid to northern latitudes, of which Ontario is included. Freeze-thaw cycles have been shown to break up organic matter, disrupt cells, and increase dissolved reactive P and soluble reactive P. Contemporary conditions do allow for a layer of snowpack. However, following along with climate change, it is also expected to reduce that. The springtime is of importance already in studying phosphorus runoff, as approximately 80% of runoff entering the surface waters occurs during this non-growing season. Excess water in these systems helps contribute to this, as excess water and wet-dry cycles can mobilize nutrients. However, evidence for soil phosphorus mobilization is incomplete. Riparian buffer zones were focused on in this study. Reason being, they are used currently as a management practice to reduce phosphorus loading into agro-ecosystem surface waters. However, it has recently been proposed more frequently that under future climate conditions with increased freeze-thaw cycles, exposure to bare soil in colder climates with reduced snowpack could potentially shift these net sinks of phosphorus to a net source of phosphorus. For our project, we looked at Olsen P, as it is a bioavailable form of soil P. We attempted to answer if there is a connection between Olsen P and the springtime dynamics of both present and future conditions, present being this field study, future being in lab. Analysis of the microbial biomass fraction was also provided in hopes to provide a new assessment of how freeze-thaw cycles affect phosphorus mobilization. The site of concern was a riparian buffer zone located in Grand Valley, Ontario. It had a catchment of 0.5 square kilometers and was filled with soil typical of this area, being alkaline, basic, and calcareous. We wanted to look at a site which received agricultural runoff and was specifically a wetland riparian buffer. These are seldom looked at, as treed riparian buffers and grassy riparian buffers are more common across the Grand River. To monitor the site through the winter and then into the spring freshet, we used data loggers to track water content, soil temperature, and electrical conductivity, as well as air temperature. We observed the changes in these properties and how this related to the behavior of the phosphorus at the site. The site looks something like this, with a drainage creek running into the Grand River, separating a wetland riparian buffer from a treed riparian buffer, which was used as reference. The wetland riparian buffer was sampled at 9 different locations in triplicate and then composited. The data loggers were located at the ovals, and water samples were taken at the crosses. Soil samples were taken from the top 10 centimeters of the soil, taken back to the lab and air dried for 24 hours. Sieved at 2 millimeters and then extracted using sodium bicarbonate solution, filtered through a Wadman 40 tube filter paper, and then analyzed using the phosphomolybdate method using the Murphy-Riley solution. In addition to the typical reactive inorganic P, inorganic fraction was deciphered using an additional persulfate digestion on the Olsen P extract prior to again analyzing with the Murphy-Riley solution. 1 liter water samples were taken at each of the respective sample sites as pointed out earlier. 6 10 milliliter subsamples of each liter were analyzed using a HAT colorimeter with the associated phosphor packets. Of initial note upon analysis of the data revealed the date March 12th of this year as being significant. On this day, it was the first real warm day of the year, So there was a lot of snow melt, and the Grand River was flooded, extremely flooded. Overbank flow occurred, and the site was inundated, resulting in a drastic increase in dissolved reactive P in the water samples taken on this day in comparison to the rest of the non-growing season. As well in this date, you can notice this is when the soil first warms. As the soil never froze truly over the winter, this is the first warming period, however. And with this came an influx of runoff and snowmelt, which you can notice here with the increased water content of the soil, and thus the associated decreased soil electrical conductivity, a dilution effect. This would indicate some form of soil flushing at the surface. On this date as well, labile inorganic phosphorus was more variable and increased on average compared to the other dates. However, this only occurred in the wetland site as the treed riparian buffer site remained constant generally throughout the spring season. Microbial biomass-P was also sampled and analyzed throughout the non-growing season. Notably, it declined from its pre-winter mass and increased throughout the remainder of the spring to recover and surpass the level observed heading into the winter. In-lab experiments were run in two different series. We used flow box experiments and jam jar experiments. Jam jar experiments were used to isolate solely the effective freeze-thaw cycles on soil, while the flow box experiments introduced a water to flow through the soil and then collect the leachate and analyze that for phosphorus as well in response to freeze-thaw cycling. All microbial biomass-P determinations were used using the Brooks et al. 1982 method with the associated equation here. Of interesting note in the lab results is that after 6 freeze-thaw cycles, there is an overall increase in labile Olsen P. This includes both the inorganic and organic fractions. However, when you break it down into experiment type, you notice that it is mostly the jam jars that are experiencing this increase in P, as they did not receive any water to flush out any readily extractable P. Here, broken down into fraction, you can see this trend occurs in both the inorganic and organic phosphorus fraction. And again, when broken down into experiment type, you can notice that the jam jar values are considerably higher than the flow box values for both the inorganic and organic phosphorus fractions when observed after 6 freeze-thaw cycles. One possible explanation for this that we observed was in the leachate. In the flow box leachate that was drawn out of the flow boxes, you notice that there is a considerable amount of phosphorus leaving the system after the first 2 freeze-thaw cycles, and then again after 6. This could potentially be the reasoning for the disappearing phosphorus after 6 freeze-thaw cycles in the flow box experiments. In conjunction with this, the microbial biomass-P was observed to decrease after 3 and 6 freeze-thaw cycles. The reduction in stability of this fraction could potentially be appearing in the inorganic and organic labile soil fractions after 6 and freeze-thaw cycles, after less of the phosphorus is being immobilized by microorganisms at this time. From the field study, we learned that upon the first flood date of the year, when things first warmed up, there was an increase in reactive inorganic phosphorus at this site in both the soil and the water. There could be a multiple of explanations for this, potentially dissolved reactive phosphorus leaching from detritus or entering from agricultural runoff upland. Interestingly though, the soil water content does spike at this state, indicating some sort of soil flush. Of notable results in the lab, we noticed that after 3 and 6 freeze-thaw cycles, there was a general increase in inorganic and organic labile P in the soil. As well, the analysis of microbial P indicated that there was a decline after this amount of freeze-thaw cycles. Indicates a longer-term reduction in this fraction's stability in the face of continued temperature cycling. This provided a new assessment of how freeze-thaw cycles affect phosphorus mobilization in the soil. In conclusion, we observed the general increase in dissolved reactive P and reactive inorganic phosphorus in the soil and water respectively upon the first snowmelt and runoff event of the spring. In lab, soils exposed to 3 and 6 freeze-thaw cycles exhibited an increased inorganic and organic phosphorus fraction. as well as a decreased microbial fraction, potentially linking these two as a decrease in stability of fractions over the course of freeze-thaw cycle exposure. Linking this to climate change is key, as you can see that increased freeze-thaw cycle exposure to the current field season could have seen even greater increases in reactive inorganic phosphorus in the system. Conditions for fine-tuning a model for phosphorus loss from freeze-thaw cycles will require longer-term data collection to predict and inform surface water quality management. Thanks to my supervisor, Dr. Susan Glasauer, Joseph Radford, Kimber Munford, and all else whom helped me through this work.